Good morning, everybody. My name's Li Jun. I'm the founder and CEO of CoJourney. So today, I'm going to share with you how to navigate the CMC journey, not only for early phase, but toward commercialization, and share a little bit of my personal journey in the CGT field. CoJourney is a global gene therapy CDMO. We manufacture GMP-grade plasmids, mRNA, and viral vectors. Top left, you can see our commercial plasmid facility, 6,000 square meter in size. We can manufacture more than 300 batches, GMP-grade per year, with our single-use systems, zero chromatography platform process, and because we're fully disposable, we eliminate the cross-contamination from the roots. We also have a clinical viral vector facility that is 1,500 square meters in size that houses a 200-liter and a 500-liter bioreactor with a fill-finish suite. There we use a high-yield, scalable, robust platform. I'm going to tell you more about that. And we're building our next commercial facility that will become operational next year. Besides the plasmid viral vector mRNA services, we also want to bring online our GMP-grade guide RNA service later this year. We also provide our QC service, full GMP QC service, and regulatory services. And our facilities are built out to be compliant with FDA, EMA, and NMPA standards. And I will share with you that we have serviced more than 40 clients and more than 300 batches. And the benefits we bring to the clients is risk mitigated, cost controlled, speedy, fast delivery timeline, and a turnkey experience. But more than anything, today I want to talk about how important it is to really work out the early phase processes that is viable through commercialization process, because you really don't have the time to fix it later. Why I started CoJourney, my journey in gene therapy field really started back in 2006 when I joined a therapy development company as a process development engineer. So this was 6 years before the first gene therapy drug, Glabera, was approved. So back then, really the field was early at its days, no scalable upstream nor downstream process. So, but later, I joined a CDMO company and had the privilege to work with many clients, and 2 of them became commercial last year. So during this process, I recognized how critical it is to have the early phase process worked out before you move to GMP to generate the early phase material that really could cut down months, if not years, in your late phase timeline. So this is a summary of 10 different gene therapies that was approved over the years. Several different viral vectors. The FDA approved is in blue and EMA is in orange. As you can tell, from BLA submission to BLA approval, there is a large variation. Some took 200 days, 300 days. Some took 3.5 years. So why is that? When we look deeper into the issue, well, some of the issues were, clinical long-term expression. However, some of the, clinical-related questions are really related back to the CMC. So 80% of the problems are with CMC. Comparability between early phase and commercial is a big one there. And, both FDA and EMA recognize the industry challenges, and they have, over the years, set out many guidances for industry as well as CDMOs. And so this is EMA guidances. So we laid out all the CMC challenges for process development, early phase and late phase commercial, and it goes across the entire drug development life cycle. So in early development phase, we pay attention to things like raw material selection and formulation development, stability testing, and that those key assays, they need to be ready to collect data to provide ground for your early phase material manufacturing. And for early phase manufacturing, the process optimization is critical because really a lot of the early decisions are made early. After phase 1, phase 2, if you want to do the change control and bridge the early phase to commercial processes, you may not have enough time to do that. So CoJourney focuses a lot of the effort in early process development and assay development. Your key assays such as VG titer, potency should be ready, if not fully qualified for your development. And FDA places key emphasis on the potency. I'll talk about that later. While I cannot go through all the challenges in this 15 minutes, I'm highlighting some of the key essential considerations, such as early phase, we should think about reproducibility, right? The scalability, how your unit operations are designed so they're not only scalable, but you want to have success in your GMP runs. You better figure out all the processes and have successful PD runs in order to move to GMP. And robustness, right? The stability of the process, the tolerance, the feasible range, all need to be figured out. The later process characterization address those issues, but the key is to push them so it's early and it's ready or somewhat ready before you move to GMP manufacturing. I want to also talk about one size fit all. Many of us think, oh, one size fit all is just not possible. But you can, for both plasmids and viral vectors, to think about one size fit all because it's not only critical, it's easy to switch from one product to another for one client or switch from client to client. So I'm going to talk about our solutions to one-size-fit-all solutions for both plasmid and viral vectors. And the other early considerations includes cost and regulatory risk and your capex for the facility, the footprint, and so on and so forth. I want to say, early days I worked with adherent process, which is not scalable, and that has a regulatory risk. And as the field progresses, we now think, early on we should have a suspension process to work with. So I want to emphasize that our journey for the clients to achieve CMC excellence is critical. Because number one, first to market, right, for any startup companies, biotech, is critical. You want to use a good platform. You want to work with someone who can take you, not only make you early phase material, but really know what they're doing. So the late phase, when you show proof of concept, is there and it's ready. We all say the process is the product, and it's not just lightly said. The process definitely has to be designed. We all talk about quality by design, but process as well. Cost is by design as well. So think about whether you want to, have the process is part of the product. I cannot emphasize the importance is part of it, right? The science is there, but the process is also critical. So some of the early decisions are summarized in this slide. Your cell line for any process that you work with needs to be commercially viable. You cannot say, I'm going to work with this RUO cells that is not clearly traceable, and hopefully in the future to fix that. It's too late if you don't have it. That decision made early. Right, the logistics supply chain during COVID, everybody, the entire world had trouble sourcing material. Right, in early development, we cannot use non-GMP grade materials and later switch down as time and as difficulty. So at CoJourney, we use GMP grade materials very early on. And the key CMC assays, I'll talk about that later. And here I want to emphasize the plasmid, for whether you're using it to make your vectors or whether you're using it in the future to do in vivo plasmid, as a DP. You need to, for early phase, use that scalable, lockdown, and commercial viable process to make your phase 1 material. And it needs to be GMP grade. Both FDA and EMA are very clear about using GMP grade and eliminate the cross-contamination risk, right? And for viral vectors, again, choose your platforms carefully. Thinking about the commercial process, think about how scalable it is, how cost-effective it is, and also use that same principle, scalable, lockdown, commercially viable process to generate your early phase material. Or you're going to fix it later, but with a lot of cost and time could be the big obstacle. So the key take-home message is plan early, plan thoroughly, and really think about clinical, but aim for commercial. So we have track records for our clients with FDA and CDI and the several approvals, and more on the way. And more importantly, we planned to cut the cost from the very beginning. So I'll talk about the zero chrome platform for our plasmids, and I'll talk about the one-size-fits-all scalable platform for the viral vectors. So we really would like to go with this journey with our clients to not only lead them through the early phase, but also support them in the future commercial. This is the most important slides for my talk. And in the top row there, you can see the industry practice, right? You do the fermentation, you collect the E. coli cells that has your plasmids, and then you lyse the cells, you precipitate out some of the impurities, and then go through a filtration, a UFDF, and then put it on several columns. Typically 3 columns to get rid of different impurities. But some CDMOs may reduce it down to 2. We have removed all the chromatography steps completely, and this has been tried in many, many plasmids. We never had to add one chromatography back. How did we do it? We plan early. We think, what we can do, push that challenge to upfront. Starting with the strain selection, the clone picking, and the banking, we optimize the upstream fermentation process to get the high yield, also high purity plasmid in E. coli. We harvest the E. coli a little bit differently, but the key know-how here is in the downstream. So we have this automated continuous process that can really not only not harm the product, meaning the supercoiled content, but also to precipitate out most of the product and process-related impurities. So after that, we do a simple filtration, depth filtration, and then put it in the final buffer by UFDF. So by doing this process, we are able to deliver high quality plasmid, meeting or beating in the most part of the 3 chromatography process. And that really brings the cost of goods to less than 70%, and we have removed all the buffers, all the bags, and it has some added benefits to the clients. This is our platform viral vector process, and this is a one-size-fits-all process. And this is a suspension. We could do either the HEK293 triple transfection or SF9 as our client prefers, and scale it up to 200, 500. And the key here is that scalability. So from 2 liter, 5 liter, 10 liter in PD lab, scale it all the way to 200. We have seen very good linear scalability as long as you really focus on the key process parameters. One of those examples is if you do your transfection well, you're going to see as high or better of the yield and also the full empty ratios in your GMP runs. Downstream, it's a 2 chromatography process, affinity plus a polishing. So one size fit all, sometimes the establishment runs shows, hey, we have a robust process, but if you need to tweak it for different capsids or for some of the novel capsids, we have observed up to 10-fold improvements in upstream, and downstream we're currently at 40% of the soup, and all the way after two purifications, we're at 90% of the full capsids. Analytical is critical to support process development. We have developed 6 different platforms, nucleic acid-based, protein-based, cell-based, and a lot of the instrument-based platforms to support development, as well as we have developed 49 QC release assays for all different moieties. So potency. It is critically important, and back in 2019, we've all learned about the later change control from animal potency for product release to a in vitro potency, how critical that was, and there was a FDA gave some warning letters to a company because the potency data was manipulated in early phase. So it's critical to start thinking about potency in early phase, and really you don't have to have the full functional assay developed from the very beginning, but think about the mRNA or protein expression, factor enzyme activity, things of that nature, and really work with FDA early. In CoJourney, we also have a QC team that is capable of helping our clients develop this critical assay, because it's needed for every step along the way, for stability, for dose setting, for comparability, and you better start working on it early on. This is my last slide. So going back to my roots, process development and manufacturing, right? So you optimize your process for early phase, however, aim for commercialization. Do more, because really after your phase 1, 2, and if you see great proof of concept and potency in patients, you don't have time to go back and fix some of the early problems, right? So building a better process, it really enhances your future manufacturing and shortens that time between early phase to commercialization. So if that 3.5-year filing timeline comes down to the normal, if we can get approved in 200, 300 days that we see for some of the products, you can help patients a lot early. So that's what we are motivated. to take on this journey with our clients. Some PD pictures and GMP pictures. So come and talk to me if you have any needs or about our process. Thank you so much.